Okay, welcome everybody to the Oxford Café Scientifique. I'm going to sort out sound in just a moment because I'm completely alone here in terms of setting up. I have a lovely bunch of people in the room, which is great, but I'm not going to ask them to do tech stuff. So if you're online and you can't hear, just put something in the chat and I will get to it in just a moment. Before that, welcome to the last Café Scientifique of the year, of 2024. We're back again in January. I think it's the 14th of January. I'll be checking that before the end. And to the people who come in person, thank you very much for turning out on a cold night. Very much appreciated. We're online as well, as usual. For those online, if you haven't been to the cafe before, then the way it works is please stay muted. Please do put any comments, questions, whatever you want to say in the chat. You can start now if you want, and we'll pick up on those once we get into the discussion part of the evening. We have a tight little schedule tonight because we have to get all our equipment out of this building by 9 when it shuts down and the employees will leave. So I will be stopping probably tonight at about 20 to 9 latest. If it's a little earlier, that won't be a problem. The way that cafe works for the newcomers, particularly those online, I think you may have several, is that this is meant to be a short, informative talk about somebody who's an expert in the area within science, aimed at the public. So there is no such thing as a silly question here. That's our motto at the cafe. Please jump in once we get to the discussion and say whatever you like. And then we take a short break for those at home to go and put the kettle on, for those in the room to refill the glasses and get more snacks, because I don't want to take any food home. So that's your mission, should you choose to accept it, is help me finish off the food, otherwise I get fatter. And then we come back, so it's only a very short break, a few minutes, and then we'll come back and start the discussion. And we tend to find in Oxford that most people want to say something to the speaker, but we can also have people saying something connected to a point somebody else has made. It's not a problem. It doesn't have to be a question. It can just be a comment on the content of the night. So I'm absolutely delighted tonight to introduce Martin Hodson. He has many roles. I'm not going to go through them all. But he's particularly associated with the University of Oxford's Human Sciences Department here, also with the Oxford Brookes. He's a visiting researcher in environmental biology, and one of the key members of the John Ray Initiative, which links together the environment, general science and Christianity. Is that right? Got it right? Just trying to make sure I remembered it. Good. I'm going to hand over to him. As usual, these talks are being recorded. This one will go out on our private playlist when I have wound down from the end of term and got time to put them all up. So over to you, Martin. Thank you.
Okay, can everyone hear me? Yes, right, good. Right, well, thanks for inviting us to speak to you tonight on the topic of phytoliths and carbon sequestration. It's now not, I think the Zoom has jammed the pinger.
Oh, that is unusual. Okay. Right, trying again now?
Yeah, we're okay. Right.
We'll get there.
Okay, right. Now, we have got a problem with carbon at the moment. The carbon cycle is out of sync, as many of you will know. We've got far too much carbon up in the atmosphere, carbon dioxide in particular, and that is causing us a lot of trouble. This is the famous Mauna Loa Observatory graph from Hawaii. And if you had the pre-industrial concentration of carbon dioxide, which is way back here, be about 280 parts per million, by the time we get to 1960, when the graph starts, it's about 315. And then it's going up by 2 to 3 parts per million every year, until we're now pushing 420 parts per million. And it's still going up. 2 to 3 parts per million every year. And that is the main cause, not the only cause, but the main cause of global warming, climate change. and why we've had 18 months in a row of above, well, record temperatures. Now, the main thing that we've got to do about that, undoubtedly, is to cut those carbon emissions. There is no doubt about it. But we can also try to take the carbon out of the atmosphere. And that's what we're going to try and talk about tonight. Now, there are a number of different ways of taking carbon out of the atmosphere, and varying degrees of success and varying degrees of danger. Off over here on the right, we've got the idea of adding iron filings to the Pacific to produce more growth of the algae, which then die and take carbon out of the atmosphere. Jolly dodgy, if you ask me. You know, what's it going to do to the rest of the environment? So you've got that kind of idea. You've got the peatland and blue carbon management. Blue carbon is things like mangroves and coastal ecology. That's really quite a useful thing to do. Then we've got enhanced weathering. There's a lot of interest in that. So putting in rocks into the ground, specific types of rocks you can enhance the weathering and get the things to take carbon dioxide up. Probably the most expensive, as you could see, is direct carbon capture and storage, which is the one that our UK government is particularly in favour of at the moment. It's very expensive and pretty well untried. You can see much more expensive than anything else. There are also all kinds of machines that people are trying to invent that are to suck the carbon dioxide out of the atmosphere with various chemicals and so on. Things like biochar, yeah, we've got a lot of interest in that at the moment. And then probably the one that you'll know most about, or you hear most about, is this one over here, afforestation and reforestation, so planting trees. Yeah? But the one I'm going to talk about is here, soil carbon sequestration. And as you can see, it's got quite a lot of, there's no really red things or horrible things here. It's to be relatively cheap, and we can do it fairly well straight away. Now, what about carbon sequestration? The first thing to note is there's an awful lot of carbon in the soil. There is more carbon in the soil than there is in the vegetation and the atmosphere put together. So that is a massive amount of carbon. And just a typical example, here's a UK forest. Notice 72% of the carbon is in the soil. It's not in the trees, it's in the soil. So it's really important. If we could actually increase the amount of carbon in the soil and trap more of it there, we could actually do something to help with climate change. So how can we increase carbon sequestration? Well, add carbon to the soil. You think of all those leaves that have been dropping the last few months, get sucked down into the soil by earthworms, broken down by bacteria, then the carbon's re-released to the atmosphere. The problems then with increasing carbon sequestration, three. One is there's a finite volume of soil. You can only add so much to the soil and then you can't add any more. The second and probably the biggest one is this reversibility thing. So you add a leaf to the soil, yeah, you've added some organic matter to the soil, but it soon gets broken down by the bacteria and everything in the soil, and the carbon's re-released back out. Then the other problem that people have noticed is that when you add carbon to the soil, it changes the fluxes of some of the other greenhouse gases like methane and nitrous oxide, which could actually make things even worse. So you've got these problems to deal with. Right, so that's an intro to carbon sequestration in soil. Now let's have an intro to Phytoliths. Pampas grass. Actually, walk down the street here and there's some pampas grass growing up. You can see it's got these wonderful, nice flowers and everything. But the thing that you know if you're a gardener and have ever tried to tackle it is it's sharp. It's very sharp. And the reason it's sharp is that it's got these little things along the edges of the leaves, and they are made of silica. They're little prickle hairs that actually really are razor sharp. If you went with a scanning electron microscope, it would look something like that. And that is made, a lot of that is silica, and that's what's going to cut you. This is actually from a wheat inflorescence, a similar sort of thing. It's what's known as a prickle hair. And those are the things that cut you when you're pulling up pieces of grass, that sort of thing. Now, how does that silica get into the plant? Well, most soils contain a lot of mineral matter, aluminosilicates. That's what they're made of. The sand, the silt and the clay is made of aluminosilicate. But that is pretty insoluble. Dissolves very slowly. But then the soluble silicon is then taken up by the plant and it's transported through the plant. We won't go into transport and all that tonight. But it's transported as a soluble silica, and then when it gets to a certain spot where the concentration increases, it precipitates out as a solid again. And that's where you get these prickle hairs and other sorts of things. And they're called silica bodies or phytoliths. And those of you that are Greek scholars, and there's always somebody in Oxford who's a Greek scholar, phyton is plant and lithos is rock. And so they are plant rocks. And there are two locations where the silica is deposited in the plant. Some of you will remember plant cells. They have cell walls which contain a lot of cellulose, carbohydrates, that sort of thing. And then you've got the actual cell lumen where the protoplast or the cytoplasm is. And so you end up with the two types of phytolith. How do you prepare these? You actually prepare them by sticking the plant material, the leaf or stem, into a muffle furnace at 450 degrees C. And so they survive, which tells you that they're pretty tough sort of things. They're pretty tough. This is a cell lumen type in the wheat inflorescence bract, and that's filled the entire cell with silica. Then these are the cell wall types. You can't see it terribly well on here, but it's pinkish kind of material here, which is the silica, very thin cell walls. Now that's deposited onto carbohydrates. So you've got the two different types of phytolith, and they have different chemistries. Now, about 40 years ago, I was working on this species, which became a kind of a model species for working on plant silica. It's called Phalaris canariensis, or the canary grass millet. And you can see it's got very pretty green and white striped bracts, the flower. But inside those are these structures, which are further bracts, right, which have got a lot of hairs on, and then the seed is inside there. So that's where the seed is. And you can see that these have a lot of hairs. And those are the hairs, higher magnification, and at maturity they are full of silica. And the reason we started studying them was that they were thought to be involved in causing esophageal cancer in northeast Iran, because they acted a bit like asbestos. And so that's them. Now I had the bright idea, these are very silicious. I thought, how do they develop? How do they actually get to like that? So I did a developmental sequence. And see, exactly 40 years ago I published this paper. So here we go. Right, here's the beginning. This is before the inflorescence comes out at the top of the flag leaf. And you can see very thin cell wall, thin cytoplasm, huge large vacuole. That's normal for plant cells. Right. Now, just as it's starting to emerge at the top of the flag leaf, this is what it looks like. You can see it's actually a bit of a miracle hit the nucleus. There's the cytoplasm still intact. Notice this black material. That is silica being deposited in the cell wall. And I could check, because if you use something called X-ray microanalysis, you can ping the beam on here and it will come up with a silicon signal. So that's as it emerges from that flag leaf. A week later, the wall has thickened up a lot and the silica is following it inwards. All of that is silica on the wall. And then a week later, it's a kind of tree ring effect. The cytoplasm starting to break down. The whole of that wall is silicified. So all of that cellulose and all of that carbohydrate is packed with silica at maturity. So that's 40 years ago. That was me. About 20 years ago, the first paper on carbon sequestration in Phytoliths by a couple of Australians, Jeff Parr and Lee Sullivan. And people now tend to associate me, particularly in this country, with carbon sequestration in Phytoliths. But it wasn't me, it was these guys. Right, they started it. And they measured the carbon in the Phytoliths and then did some global calculations and reckoned that the accumulation of phytolith occluded carbon, phyto-C, within soil is a process, it's actually important there, process in terrestrial sequestration of carbon. So they reckon a lot of carbon getting locked up in these phytoliths. But it got more complicated. The method that people used for preparing the phytoliths was actually either dry ashing, 450 degrees C, or very hot acids, that sort of stuff. And then you take all of the carbon, all of the stuff from around the cells out, and you hope that there's something come up in the middle of it.
Oh. Sorry about that.
Yeah. And you hope that you've got the right amount of carbon still left in the phytoliths, which you can then analyse. And you can see that the results vary. I produced one of the papers that our group did. These guys got into a big argument about it. Because essentially, if these guys are right, if Parr, Song and Co are right, it's really important, the carbon sequestration. If these guys are right, then it's not important at all. Now, all of these measurements using these techniques, all of the measurements are on a mixture of phytolith types. So, it's a lumen and the cell wall and everything. And I have to say, what is the relevance of acid and high temperature and all of that to what happens when a leaf falls on the soil at room temperature and pressure and not into concentrated acid? What do these measurements really mean? Now, how much carbon is there in a cell wall phytolith? This is quite interesting, because around 40 years ago, there was a lady called Carol Perry who was working in inorganic chemistry in this very institute. She is now the Professor of Inorganic Chemistry at Nottingham Trent University. And she was doing her PhD on it, and I came and visited her, and she started working on these hairs. And you see, she could pull off those hairs. You didn't need to do acid digest to get them out. You just pulled them off the bracts and broke them off, and then analysed them. 40% silica, 55% polysaccharide, and 5% protein. And then I started, it was about 6 or 8 years ago, I started working on carbon sequestration in Phytoliths. And I thought, that's a lot. I wonder what concentration of carbon that actually is. That's 25% carbon. So it's a lot higher than all of those estimates that we'd seen before. A lot higher. So I thought, we need some other methodology to have a look at this. So how are we going to do it? It turns out I've got to the kind of age where I have all these younger scientists who say, Martin, can you come and help us, please? And it's quite nice, really. So I say, oh yes, I'll come and help you. So Felix de Tombeau is a French guy who works, young scientist, about 30, he works in Montpellier, and he asked, well, we were asked to put together this paper, and he got some friends in Australia who got the most amazing kit for doing microanalysis. Yeah, the kit has got a lot better than my sort of 40 years ago, because 40 years ago we could measure the silica, but you couldn't measure carbon because it was too low in atomic weight. But now we can measure the carbon as well. And what he found was, these are Phytoliths here, he found, when we did the analysis, that there was 16% carbon in these Phytoliths using this microanalysis, which kind of confirms the work that Carol Perry did all those years ago. So it's now two methods which suggest much higher carbon in these Phytoliths. And moreover, our friends from Australia they've got some amazing kit. They managed to cut a section through one of those Phytoliths, and then to do the first ever simultaneous silicon and carbon analysis of a section through a Phytolith. And so the carbon is in purple and the silicon is in green. And you can see, you imagine bacteria trying to get in amongst that lot. It's going to really be difficult for them to get there. This is nanometer scale sort of stuff. So we know there's a lot of carbon in these things. So, summary of non-heat acid results. Right, so we've got the Phytolith cell wall, 25%. The sedge conical Phytolith, 16%. Didn't mention the outer walls, that's even higher at 40%. Rice lumen Phytoliths we did for a comparison, 2.5, but we'd expect that. The lumen Phytoliths are much lower, they've not got a cell wall base to them, but all of those a lot. So carbon in Phytoliths is variable, but it can be high in the cell wall types, and values are much higher than those heat and acid preparations. So there should be a lot of potential for carbon sequestration. Yippee! But, there is a big but. What happens when they go into the soil? The snag is that Phytoliths are not formed of kind of aluminosilicates and crystalline material. They're formed of amorphous silica, which is known to be more soluble than the crystalline types. And so particularly the smaller bits get dissolved pretty quickly when they get into the soil. So we know that it's more soluble. So you think, oh dear, that's going to wreck everything, everything going to be dissolved. No, not the case. Because one of the other areas that I've worked in a lot is archaeology and paleoecology with Phytoliths. Again, people ask me, can you come and help us with this? I'm working with a group in Brussels at the moment who are working on Phytoliths. Now, you can't see this very easily, but what I did was I went to the archaeological and paleoecological literature, and I didn't have to do a major survey, it just all popped off the pages. All of these are examples of cell wall Phytoliths found in samples hundreds and thousands of years ago, which suggests that a lot of them can survive that sort of length of time. So they are surviving. What about the ideas for the future? How are we going to actually how we're going to actually use this. First idea is, what about using what's called conservation agriculture? It is a fairly modern technique where after you've harvested your wheat or rice or whatever, you allow all of the stubble and the leaves and everything to sort of stay on the surface of the soil, to rot naturally, and of course then the phytoliths would be joining back into the soil. So that's a pretty good idea. We could breed for plants with higher amounts of carbon in phytoliths. There is some evidence that that is the case, that there are variations in the amount. And then there's this rather amazing idea of burying plant bricks. A year ago, believe it or not, in the Washington Post, I spotted this article. There's a small company in the States, in California, that have invented a system for producing bricks of plant material and then burying it underground to remove the carbon dioxide. And critically, they can do it for less than $100 per tonne, which is the absolute, gold standard. We want below $100 per tonne. I've been in touch with the guys who did this work, because what I think they need to do is they need to use silicious plant material, because I think their bricks will last longer if they use silicious plant material than if they don't. So you can see there is a possibility there. So what have we got to do? It's only really the last year that we've really got started on knowing what the real concentration of carbon is in phytoliths, I think. We need to know a lot more about what happens in the soil. We're having discussions with a modeller in France who's into modelling these kinds of things in the soil. We need to know how quickly these things break down. Then we've got to work out how important this is. In our paper from earlier this year, we did calculations to try to work out how important it was globally. And the snag was, using the lowest possible results, it's really bad. But using the best possible results, it's really good, for the dissolution of the silica. So it varies depending on whose results you actually put into your equations. So it's a bit of a problem. We don't know how important it is yet. And then, and it's probably not going to be people like me and Felix that do that. It's going to be, how are we going to use that? So it is going to be the people who are producing these plant bricks. It's going to be agriculturalists. It's going to be that kind of person. But I think you can see that there is some potential for taking carbon out of the atmosphere using phytoliths. Thank you.
Thank you very Thank you very much, Martin, indeed. We're going to take a very short break now. It won't be for very long. In the room, I'll stop recording for a few minutes. Please go and help yourselves to everything on the table and come back in a few minutes. Thank you. Definitely go down that.
Okay.
Good. Okay. Back again, everybody, because I am going to stop you 5 minutes early tonight, so we might as well get on. So we have some questions and comments coming in on the chat online. Please do carry on with that. I will be reading things out. But we always start with people in the room as soon as anybody wants to kick off. Yeah?
Yeah, I mean, obviously the plan here would be to grow this on a mass scale, would it?
Yeah, could well be.
Well, I mean, instead of wheat fields, it would be
No. What we could do, you see, which I think is really good, is we grow wheat, we harvest the grain and the seed, and then we allow the leaves and the stems to rot. They've got all the phytoliths in, so we sequester the carbon into the leaves and the stems in the phytoliths, but we still have food to eat. The same with rice. So we're not saying we're going to have to grow different crops, we're just saying we might have to do different management of those crops.
So plough it back into the field.
Yep.
Right.
Okay, let's go online then. So going right back to when you first started describing the phytoliths, does fern have similar edges to its leaves? I suspect this is a gardener who's got to end up in bitten by a fern.
There are a lot of things other than silica which can cause quite sharp edges. Actually, but ferns do contain, some of them do contain quite a lot of silica. I actually did do some work on bracken, and bracken, the stems of bracken have a lot of silica in when they're mature, and they can really cut your hand when you try to pull them up. So yeah, some ferns are pretty heavy silicon accumulators.
So some of our natural kind of moor habitats are actually doing double the duty for the environment. Fewer people living on them and more silica as well. People who come to this a lot will know that I am actually by trade a chemist, and therefore I have to ask you, the word silica in chemistry means SiO2,
Yep.
doesn't have any carbon in. So what is it that you're referring to here? In what way does carbon get into it? Do you know the formula?
Now that is actually getting quite interesting just for the moment. The standard sort of thinking for many, many years was that the silica and carbon more or less were separate from each other, and that they certainly didn't bond with each other. And so when you see those pictures where you've got the silica and the carbon, the standard thinking was that the silica and the carbon were kind of separate. So SiO2 would be separate from any of the carbohydrates.
So carbohydrates, not carbonates.
Yep, carbohydrates. Okay. But now, the thing we have never found in nature is the SiC bond. You can find it in things, your shampoos and things like that, but you can't find it in nature. But over the last about 10 years, people have started increasingly to find SiOC bonds in plant material. And so I suspect that it is actually, some of that carbon is being bonded to the silica. But that's very recent stuff.
That's very interesting. I mean, the reason why you don't see SiC in nature is that it's a very weak bond.
Yeah.
Unfortunately, nothing that makes a strong bond works between those two elements. But SiOC would make a lot of sense. So you think that that's silica kind of grafted onto carbohydrates or vice versa?
Yeah, I think it's carbohydrates.
On the outside, it looked like in that picture.
Yeah.
Okay.
The only thing I know about silicon is that diatoms have a lot in it, and there are a lot of diatoms in the oceans and the fresh water. Has anyone looked at a similar thing in diatoms?
There is a lot of research on diatoms and silicification. I've been to conferences where they've spoken a lot. You'd have the plant person like me, and you'd have the diatom person doing their talk on silica deposition in diatoms. And they do take some carbon out of the atmosphere into they have these really ornate frustules, and they do have some carbon in there, and when they die, they go down to the bottom of the oceans, and they do take some carbon out. And that's one of the thinkings behind that whole fertilizing the oceans bit, because if you fertilize the ocean, you get those diatoms and other things like that to increase photosynthesis. When they die, they take the carbon out with them. So yeah, diatoms, a lot of carbon there. Yep.
saw you twitching.
Oh. Rachel always likes a silly question. So, what is carbon in the context of this illustration?
What is
Carbon.
carbon in the context of...
What sort of carbon have you got?
What sort of carbon? It's going to be things like carbohydrates. It's going to be, there's going to be quite a, in the case of plants, there's going to be a lot of lignin, all those kind of things. A whole lot, some proteins, not a lot, but it's going to be more carbohydrates and lignin as being the two big ones, and phenolics.
But I'm thinking in the context.
Hang on.
I'm thinking in the context of, are you capturing CO2 or can you be capturing methane?
We can, with this methodology, you're only going to capture directly CO2 because we're talking about plants photosynthesizing and the carbon then getting trapped in the phytoliths. However, methane is a very powerful greenhouse gas, as you probably all know, but methane breaks down pretty rapidly in the atmosphere, unlike carbon dioxide. And when it breaks down, it produces carbon dioxide. But plants don't take up methane.
Right, just going to the back of the room, then I'll go online for a bit.
So a lot of the slides were about kind of... Oh, okay. I'll hold it like this. So, like, you covered like technological advancements, so the role of scanning electron microscopy. So I was just wondering, if you're on the future research side, if you can see any like challenges or opportunities of future technology in terms of this research.
Future technology. A good question. Yeah, you can tell one of our students. A very good question. Future technology. I think, as I kind of mentioned to you, the modelling of the silica in the soil, I think, and that's going to require mega computers to do it, really. To do it properly, we're going to have to, it's going to be almost as bad as trying to model the atmosphere for climate change, trying to model what's happening in the soil with all these bugs in there, all the different chemicals and whatever, trying to work out what's happening there. I think that's our trickiest bit now, because I think the technology on the analysis of the phytoliths has improved markedly in the time I've been working on it. So we can measure carbon now in cells, whereas we couldn't do that before. But I think the actual technology for working out what's happening in the soil, we haven't got it at the moment. Not good enough anyway.
Quantum computing is all I'll say to that.
Yeah, I think so.
Okay, I'm just going to go online for a little bit then. So, do we know anything about the genetic control of phytolith formation and how this might be exploited to develop genotypes able to sequester more carbon?
Genetic control. Yeah, well, there's quite a lot of interest in it. Let's take an example of maize. Right? Maize phytoliths are used a lot in archaeology. Maize as a plant doesn't actually exist in the wild. The only thing that exists in the wild is a weedy little thing called teosinte, which is in South America. And the teosinte has phytoliths in it, and so does the maize. But when the maize is a big polyploid, it's suddenly emerged. There's a miracle that happened. And when it happened, people were thinking, wow, you're going to get this massive amount of grain, whereas the teosinte is a tiny little thing. But what happened at the same time was that phytoliths massively increased in size. So it's the genetics there was it actually they got bigger. The same, I had a student up at Oxford Brookes years ago, 30 years ago, I guess. She worked on wheat. Again, wheat is a very complex mixture of weedy grasses that have been brought together. The ancestors have much smaller phytoliths. But as you get to these multiple complex efforts that we've now got, triticum, they get bigger. So, you know, there is definite control of phytoliths, at least for size. Yeah.
Seems like you need some interdisciplinary collaboration on this.
Oh,
to
well,
get everything right.
the great thing about phytoliths is there's a huge amount of interdisciplinary.
Why do the plants want the phytoliths? And also, is that why do they scale up? In the case of those two plants, why do they scale up as they get bigger?
Why do the plants want phytoliths? Right. This is what's known as teleology in the trade, but anyway. Defense against pathogens, against grazers, all that. There's no doubt there's a lot of in the literature about them being a good defense. The other thing is that for grasses and cereals, they actually help things stay upright. Yep. So strengthening. Yeah, so strengthening and defence are the two main things, I think. Now, what was your second bit? I forgot the second.
The scaling.
Why?
So why do they get bigger in the case of the maize and its ancestor plant?
Right. Very simple. The cells get bigger. As you go towards a polyploid, the cells get bigger. So the cells get bigger when you put the silica in it. The silica gets.
So if it remained not single ploid, then it wouldn't have got bigger.
No, it wouldn't have got bigger. No.
You managed to ask a question to somebody online, unfortunately. So that's going to be good. Okay, let me move down a bit. So, do you work, Martin, with the soil sections of Cranfield and Reading and other agricultural colleges to create wider awareness?
No, I'd really like to.
If you've got this is to Chris. If you've got an introduction, Chris, send it over to me and I will pass it on to Martin.
Yeah, I'll tell you the truth. The snag with this research at the moment is it tends to be a little bit in a bubble. It's a bubble of people like me that are into phytoliths. There's a new thing we're talking about it called Blue Sky, which is the Twitter replacement, where all the scientists have suddenly migrated in the last 2 weeks because they don't like Twitter anymore and they're getting off it. And so they're all there, and the phytolith people are there. But there's a community of people, there's archaeologists, all kinds of people, and we all talk about this all the time. But getting into the main body of soil science has proven really difficult. Yeah, really difficult. Yeah.
Hang on. Wait.
I've got an allotment in South Oxford. The horsetails grow very well on it. I'm pretty sure they have silica. Horsetails.
Yep.
Would that be planted to grow using your methodology and theory?
Yeah. Horsetail equisetum is one of the heaviest silicon accumulators on the planet. So it would be a potential plant for doing things with, but you can't actually eat horsetail, which is one of the problems. Yeah, but I think that there would be advantages in growing wheat and be able to harvest the grain and then do the silica, rather than having to grow lots of equisetum and not be able to eat anything. So I think that's probably the answer to that. It does accumulate a lot of silica. There's been a lot of interest in it. It's great.
So it both accumulates silica and also creates actual phytoliths, because
Absolutely.
you won't always get both, will you?
Yeah, it accumulates a massive. If a plant accumulates a lot of silica, they're bound to have phytoliths. They have to.
I'm happy to donate mine in my garden to research, should you ever need any. Surely this would be the kind of thing that the Lego brick company ought to be thinking about. Because if there's something you want to stick down the bottom of a mine or in the ocean and bury for thousands of years, then it would be horsedown, right?
Yeah,
Any gardener would know.
yeah, it'd be fantastic. I mean, yeah, the Lego brick people would, yeah, they should be to it. I mean, but I have told them, I mean, the other big one that they could do, which we have a lot of, is rice husks. Rice husks are some of the heaviest silicon accumulating thing. You can get 20% dry weight silica. It's a massive amount. And of course, we get lots and lots of rice husks because they're a waste product of growing rice.
So have you done the carbon measurement on those husks? Do you know what the carbon storage would be?
No. I mean, it's still, yeah, we've measured the lumen Phytoliths because we wanted to measure a lumen Phytolith and they're low, but for sure the cell wall ones will be high.
Next job then.
Yeah, next job.
Okay, keep getting my attention in the room if you want to. Let me carry on then. So, is there anything linked to that? Is there anything perhaps people without allotments as well could do in their day-to-day lives to make a difference, perhaps in the gardens? Should we be all planting pampas?
I'm not sure I would all be planting pampas. I think, to be honest, in gardens, I think the less digging you do, probably the better. Now, this will be good news for some of you who don't really want to do digging, but the more digging you do, the more you expose the soil to the atmosphere and the more the carbon dioxide is released. So if you wanted to do a minor thing to help save the planet, then probably that's
And what about just gone through the main kind of bulk of the leaf dropping in the autumn?
Yeah.
And they're all lying on our lawns at the moment if we haven't swept them up. Should we be leaving them so that they can be pulled down into the soil by the earthworms? Is that better than sweeping them up?
Yeah. Definitely you want them under the ground if you can possibly get them there. Yeah. It's actually one of the very first things I remember as a scientist. I was at primary school and the teacher said, "I want you to get your parents and go out with a flashlight," it was the autumn, "and see what happens under the trees with the leaves." So we went out, my mother went out with this flashlight, and you could see the earthworms drawing the leaves down into the soil. And I've always remembered that. I was doing about 6 or something at the time. So that was the first bit of soil science that I did. And it's kind of stuck.
I'll come back online in a moment, but I'm just going to ask you a slightly more kind of difficult, perhaps, question. I think, if anything, the modern generations now are aware that we have to think about consequences. Maybe our predecessors weren't as good at doing that, and we're paying a price. Has to be a question. You've talked already about other kinds of balance, so affecting methane. Is there anything you can think of that might be a problem in terms of bringing more silica perhaps into the soil? Or indeed, if it were buried, are there any obvious problems that we should be worrying about trying to use this in a way? So silica's attracted carbon, could it go wrong?
I can't think of anything at the moment, to be honest. It's pretty natural in the soil anyway. You're just increasing the amount of it.
Does it have an effect, for instance, on the amount of moisture in the soil or not?
Might do. Yeah, I mean, that's why we need the agriculturalists to start coming in and looking at it more seriously. Because if we start doing that, then it is going to have consequences. But at the moment, I couldn't say, because we're still at this kind of, well, it's this guy with a lab, isn't it? It's not really got out into the field much yet.
Makes sense. Going a little bit more into the kind of science of it from somebody online, a couple of questions kind of related. So I was going to put this another way, but effectively what form does the silicon derive from? Is it kind of from sand? Where does it actually come from? And my question added on to that is what type of silica is it? So you said amorphous, that sounds a bit more like sand form, which of course means it erodes quite quickly. And also linked to that, how does the silicon get incorporated? Is there anything known about which enzymes maybe in the plant are involved?
Yes, right, okay. The silica, right. In the soil, largely, most of our soils in this country are what are called mineral soils. There are a few organic soils like peat and so on, but mostly they're mineral soils. And most of those consist of aluminosilicates and things like quartz and so on. And they form as sand particles, the bigger ones, the silt, and then the clay, which is the smallest of them. Now those all dissolve very slowly because they tend to be crystalline. Now, actually, one of the major sources of soluble silicon is the biogenic silicon, including the phytoliths, not just phytoliths, but other things that grow in the soil. So we've already said that they're actually fairly soluble stuff, so they actually dissolve to produce the SiO2, which is then taken up by the plant. Now, what was about 18 years ago, a guy we know in Japan, Jianfeng Ma and his group, working on rice, discovered these silicon transporters. There are special enzyme type things in plants, particularly in things like rice, that are very good at sucking the silica in and concentrating it. And he's worked on these things for years now. And we know that they're in an awful lot of plants. And in fact, but there are some plants that don't take up much silicon. And we think that basically a whole lot of them have got the transporters, that they're just switched off. So yeah, there are transporters that take up the silica. He's recently, last year, he found a transporter which seems to pump the silica towards the phytoliths in the leaf. So, phenomenal stuff. It's all molecular biology, and so well beyond my pay grade.
Good. Anything else in the room?
You probably covered this, but can you just recap on the importance of the silica in this whole topic?
In what way?
Well, without it, you've got a standard tree or root or something. So, how does these silicate structures improve matters?
Well, as I said, it's they are involved in defence because of being very sharp and so on. The defence. Actually, if you've ever got stung by a stinging nettle, that's silica. Little hypodermic syringes that actually inject you with a poison, and they're made of silica. So they're a defence. They're also involved in strengthening the plant. For most plants, it doesn't look like silicon is an essential element, though. So you can grow the plants in water cultures without adding the silica. There are a few which, equisetum actually is one where if you try growing that without silica, it flops over and dies. Rice is another one. If you try growing that without silica, flops over and dies. But the majority of plants, you can grow them without silicon. You couldn't grow them without potassium, nitrogen, phosphorus and all of those things, but they can grow without silicon. But they need silicon in the field to defend them against all these pathogens and bugs that are going to attack them and all that kind of stuff. And for keeping things like wheat and barley upright. Yep.
Not quite what I meant. And more in terms of carbon capture, what's the advantage of having silicon involved?
In terms of carbon capture, the advantage of having silicon involved is that we think that when the phytoliths go into the soil, they're not going to be broken down anywhere near as easily as just your average organic matter, which is in the soil. You had a question about...
I was going to say, what about big trees?
What about big trees?
I know some trees do have a lot of silicon in them, they're a pain because they wear your saws down.
Yes. Bamboo. Bamboo is absolutely chock full of silica. But actually, there's a very interesting balance, because most trees use lignin to keep upright. That's a
And water.
polyphenolic type thing to keep themselves upright. And there is, in fact, I've been writing about it recently. There is a thinking silica should be cheaper to use, but the thought is that it's quite a lot heavier than lignin. Then you see the equisitums in the equisetum ancestors many millions of years ago were much higher, and they grew much. They were like tree type size, or moderate tree size. But lots, 30 metres or so. But when trees got really going, when your lignin started to get, they outcompeted them because they could go higher. And the thinking is that the silica was heavier than the lignin, and so the boughs would drop off, and they couldn't hold themselves up high enough before they just fell apart. Whereas lignin is much lighter structure, you can go much higher with it.
There's more variables in that because the conditions on the earth have changed since those days, so there's more going on maybe. The other thing that I think will be interesting is that it could be the other way around, just going back to your point a moment ago, it could be the other way around, that for the plant, having carbon involved in making the phytoliths is a better way for them to capture silicon and use it structurally. It may be better to do that than just have silicates on their own, which I suspect that kind of lack of solubility is an issue there. You can't get, if you're trying to absorb silicon from quartz or sand, it's difficult to get it into your system without making it soluble, which carbon can then do.
Yep. There is some literature on that where they put out acids from the roots and things like that, which are involved in dissolving the mineral matter around the plant. Yeah.
And the business about being good against pathogens is, of course, because silicon isn't used much in kind of the pathogenic, the mammalian, the insect world, a little bit more in the insects, the exoskeletons, but it's not really part of making a movable living system function.
And actually, with the pathogens, what actually happens often, the pathogen tries to penetrate the leaf, and around the pathogen, the silica gets deposited, so it forms a physical barrier against the pathogen going any further.
I've got a nice one online. Can phytoliths be produced in the lab with enzymes?
Not at the moment.
I mean, could we end up maybe with some kind of arrangement on roofs of houses which are just quietly growing phytoliths and then turning them into some source we can bury?
Not at the moment. I think we're in the realms of science fiction at this point.
Grow wheat on your roof.
You can grow wheat and you can grow all of these other plants, but actually growing phytoliths in the lab, no, not no.
Not yet, anyway. I mean,
No.
things change, don't they? So, yeah, we'll see. And then another one line. How efficient in yield is the carbon sequestration in phytoliths compared with other plants? I presume that's not taking into account how long they might also last before decomposing, but just straightforward yield.
We don't know. I have to be honest, we don't know. mean, the suspicion is that they're going to last longer because as you see, it's all protected by the silica. And so compared with just the carbon, just the leaf material that is not silicified, that's more likely to rot easily. But, you know, putting figures on it, at the moment,
What about, say, the carbon load of an average plant that's like a phytolith, but is, you know, something similar, but so if you go from low silica versus high silica, where does the carbon yield change there? In terms of what it's captured. I mean, that would be an interesting thing to find out.
I mean, don't know. I mean, the thing with a scientist, the ones who say they don't know, I think you have to be more trustworthy than the ones who just say they know the answer to everything.
It's quite rare we have an environmental biologist here, so I'm just going to ask you for the last kind of few minutes to think about what else you think, moving away perhaps from phytoliths, more generally about the environment and the way that biology works in it. What should average people be thinking about, trying to encourage, maybe doing for themselves to help improve the planet, in your views, with that specialism?
I think undoubtedly we have to think about climate change. You just have to think about climate change. It looks... I mean, I do a lot of talks rather like this, not such on a specialist topic, but you know, to general audiences and all that kind of stuff on climate change. And every time I put one of those talks together, it looks worse. It really is getting bad. And for those of us of my colour hair, you know, we probably won't be here to see it when it gets really bad. But if we're not very careful, we could have a totally wrecked planet. You know, and you know, I do all these tutorials, as at least one of you knows, in human sciences about this sort of stuff. And you know, at the moment, we haven't lost very much biodiversity strictly because of climate change. There's been a lot of movement of animals and plants, you know, further north in the northern hemisphere where it's cooler, or up mountains, that sort of thing. But we've had 1.2 degrees sea rise above pre-industrial. If we get 3 degrees by the end of the century, then we're going to lose an awful lot of our biodiversity. You know, I mean, I got into this actually through a very famous Jesus College scientist, Sir John Horton, who was the first chair of the science panel of the IPCC, and I worked with him for quite a few years. He sadly died at the beginning of the COVID pandemic. You know, his words from years ago, you know, climate change is a weapon of mass destruction. And, you know, he's right. You know, and that was 20 odd years ago, he was saying that kind of stuff. But they still haven't done anything about it. You see all of these meetings we've had recently. The 2 COP meetings. We had the biodiversity COP, where everybody was walking out and there was no real agreement to do stuff. You had the climate change COP, where you got a kind of an agreement, but will anybody pay any money into it? And then the plastics just went totally to pot. So we've got all of those things that are happening, and you kind of think, we've got the technology, we've got the sort of stuff to do to solve the problems, but it's the political will which is missing globally, not just our country, but globally. And then I won't get into it. But you know who's going to be the next president of the United States?
I think we can brainwash him. I think it's eminently brainwashing. Get him to see our point of view. I'm also going to ask you, so as you said, I like a silly question, but I'm going to ask a silly question of my own. One of the things that has interested me for a while now is this idea of doing things like kind of pumping carbon underground into old gas fields, oil fields, things like that. There are issues. It's a little bit like the fracking problem. You take stuff out of the Earth's crust, and there are spaces that can cause more disturbances, more problematic. This seems a bit kind of left field, but going back to kind of using phytoliths in the right way, because you were talking about plants that have very high concentrations of silica in very small bodies, things like little spines on their seeds or their husks or things like that. We're not talking about it needing to be long fibres. You know, could you imagine there's something where this was used as a vehicle? Because if you're going to take an empty, cleared out oil field, you need to put something with structure inside it. And this would feel like the right kind of thing to maybe push down with some water, and the water will seep away and it'll deposit the silica. That kind of a solution kind of marries the two bits of technology, doesn't it?
That's kind of like burying the bricks, only much further down.
You can get into smaller holes if what you're taking down there is a suspension of bits of rice husk in water.
I think it sounds a sensible plan. It's a lot more sensible than what the UK government is currently proposing, which is the standard carbon capture and storage, which is that you go ahead with building your power plants and then you capture the carbon dioxide and you pump that underground, and then you hope that it doesn't escape later.
Gases have a habit of escaping.
Exactly. Then the stag anyway. They're thinking of spending 20 billion on that. 20 billion, we could do so much in terms of renewables, in terms of everything else in this country if we did that. But 20 billion on that technology, when we don't know that it's going to work at scale, and when it's probably going to take 25 years to actually get it to work, we have not got 25 years to do that. Daft. But your idea, I think you should get it. I think the next Nobel Prize is that one.
You need to think of silly ideas and try them. Although I get the problem that governments are in. If you don't start doing something, you will be 25 years on and still go, we don't know if it'll work.
Yeah.
We've all been in institutions that play that game. So yeah. Okay, if there's nothing else, then I am going to close up. Give me a nice long time to clear up, which will be good. Before I say thank you to Martin, a reminder that the cafe mailing list goes out, excuse me, regularly before meetings, tell you what's going on. We have the next cafe is coming up on Tuesday, the 14th of January, and the speaker is Claire Grimond, again from Oxford, talking about something completely out of this earth, literally. She researches both finding exoplanets and how to determine what their atmospheres are. And the older and more grey-haired amongst us remember that when we were young, there was no idea there would ever be planets of Earth-like planets elsewhere, because the technology for detecting them is just not there. It's been a revolution in the last kind of 15 years or so to discover how many planets there are in other solar systems. And of course the interest is whether those might one day have a form of life on them, or may have had one in the past. So Claire's going to come and talk about that to brighten up our new year, which I think should be fun and very different from the quite, biological Earth-based stuff we've been doing in the last few months, just through a trick of the scheduling. For anyone who hasn't seen it already, if you Google Oxford Café Scientifique, you don't have to put the accents on, but you can if you want, then you will find our website, and at the bottom there it says how to join the mailing list. There's no requirement to turn up regularly or anything like that. I think last count we had 875 people on the mailing list. We do not have 875 come to meetings. So come whenever it excites you. Second Tuesday of every month except for August, because very early on both me, my co-founder, and our regulars decided we needed a break. So every month except August. And this year, thankfully, the February one does not fall on Valentine's Day, which is good, because every few years it does, and that's more difficult to get a speaker apart from everything else. So please do sign up. You can unsign yourselves any time. You don't have to have an Oxford email address, it's open to anybody. If you're online, the links go out always at 2 or just after it on the afternoon of the day of the talk. So you need to sign up to the mailing list before that point in order to get the link. And then videos will go on. I will be doing an email when I have put those up. You can see we've had some very good ones in the last few months, quite a few of which will go on the public link as well. The private links are a privately unlisted YouTube channel playlist which you can get to the link. I'll move away from the dead spot. And then the public YouTube channel anyone can get to and look at. And there's a few of the speakers were very happy for it to go on there, so you're welcome to share that link around absolutely anybody at all. I may be joining Blue Sky, haven't decided. I am still on Twitter with this, and we're still on Facebook as well, and occasionally to mailing lists in Oxford, but I've never seen any big improvement in numbers from that, so we will see. But we will be back in the new year. In the meantime, everybody have a happy Christmas, and thank you particularly, I'll give you another round of applause, to Martin for tonight. Turn the recording off.